Monday, but the following Monday. And format's going to be very similar to that. On test number 1, as I've already mentioned to you, the exam will open up at 5:25. So try to be here early. Make sure you're ready to rock and roll. Where did I put this in? I put out an announcement, I believe, earlier this afternoon, and I thought I was going to put it in the outline here, but I guess I didn't. Next Monday night, we're going to do a practice quiz in here. We'll do that at the end of class. And just to make sure that everybody's able to go. Make sure everybody remembers their login. And hopefully no problems there. I just want to emphasize that the information in the electrophysiology lab and electrophysiology homework, they're supposed to be complementary of one another, and you'll see the same types of topics and issues being brought up there. And make sure you're familiar with the rules that are included as part of both of those, actually, both of those. I posted some information out there that I shared with you on Monday night regarding the GLP-1 agonists. So just to be clear, GLP-1 is a naturally occurring molecule. The GLP-1 agonists, like the Ozempics, the Mounjaros, the Wegovys of the world, those are modified molecules. And those are basically synthetic, and the key thing is that they've been altered so that enzyme DPP4 cannot act on it, and therefore the effects of the GLP-1 are long-lasting, not short-lasting. And in that context, in one of the files that I uploaded, you'll see this type of information here, which is just kind of a summary of what I showed to you. And so here in the far left-hand column, you can see how GLP-1 works in our body. And here it basically describes exactly what I mentioned to you. Talks about GLP-1, and then it talks down here, you can see this kind of light red colored oval that talks about DPP4, and it says that this is an enzyme that removes GLP-1 from the body, and as a consequence, you feel hungry now. And then over here to the right, you can see that it's talking about this class of molecules, semaglutide. This would be Ozempic, and you can see here it says it's resistant to DPP4. And little bullets here say, doesn't get removed as quickly as the naturally occurring GLP-1, and this makes you feel fuller for a longer period of time. That's with regards to the control of hunger. But remember, this class of molecules was really developed to help treat diabetic patients. That's the critical thing. Anyways, make sure that you look at this and you got that information in hand. And then just to remind you, there are those articles on diabetes, just to reinforce the theme that diabetes is typically a disease associated with inactivity. And for those individuals who are regularly active, their risk of developing diabetes is decreased significantly. A different way of saying it is that their hazard ratio, or their risk ratio. Remember I mentioned to you that in epidemiology land, they'll use the term hazard ratio. If you have a hazard ratio of 1, that means you're at equal risk for the general population. If you're above 1, that means you're at greater risk. If you're below 1, less of a risk. You can use RR, relative risk. Anyways, those terms, all they're trying to do is describe a population and how a particular intervention affected the risk of developing a particular disease, whether it's diabetes, hypertension, melanomas, you name it. All right. So we're in this area that I refer to as cell-to-cell communication. and hopefully you learned a lot about how it is that cells can talk to one another. I'll just say what I mentioned Monday night, and I said previously, and that is glucose homeostasis is a major theme of my class. Why? Because diabetes is one incredible health issue that we're dealing with. It costs billions upon billions of dollars every year. The cost of healthcare in our country is absolutely insane. And that's why I said maybe we need to go to China and see how they deliver healthcare. All right, so with regards to neurophysiology, we've talked about electrophysiology, and I want to come back to a point that I made last week, and it's regarding the conduction of action potentials, and this is going to feed into something that we've already talked about. So I'm going to get into that, and then once I'm done with that, I'm going to come back and we're going to start talking, or I'll continue talking to you about the events occurring at the synaptic cleft and how it is that we produce either excitatory or inhibitory postsynaptic potentials. So that's the game plan for tonight, and there's a lot to discuss. So using this diagram to your left is kind of a synopsis of our discussion with regards to action potentials. Here I'm representing the axon. We've got the axon terminal down to the far right there. But in the middle, I've got the propagation of the action potential. And last Wednesday, I was describing for you the key factors that determine the speed at which the action potential can spread down along the length of the axon. And we use this term, conduction velocity, to make reference to the speed at which the action potential spreads down along the length of the axon. And if you recall, one of the points that I made to you is that in large myelinated axons, the conduction velocity might range somewhere from, say, 60 to 100 meters per second. And whether you can grasp that or not, whether you can visualize that or not, and I hope all of you can. Maybe I should make a homework assignment that everybody watches football this weekend. And then you sit there and watch these football players running down a field that's 100 meters in length, and imagine somebody doing it in 1 second. That's what your axons can do. It's incredibly fast. So in large myelinated axons, the conduction velocity is somewhere around 60 to 100 milliseconds. In small unmyelinated axons, the conduction velocity is a lot slower. And here we're going to say that it ranges from something like about 0.5 on up to maybe approximately 5 or 6 meters per second. Much, much slower. And one of the points that I made is that the presence of myelin acts as a conductor, and it allows the action potential to skip down along the length of the axon, skipping from one node of Ranvier to another. Remember, in myelinated axons, that's where the voltage-gated sodium ion channels are. The voltage-gated sodium ion channels are not found beneath the myelin. They're found at the nodes of Ranvier. So the action potential skips down along the length, that type of conduction we refer to as saltatory. That's what the word saltatory means. It means skipping. But I also mentioned that the radius of the axon is important. And here's a, we'll say here is a axon with a relatively small radius versus another axon with a fairly large radius. This comes back now to an important thing that you learned previously. And here I'm going to kind of tip this off a little bit, is that the leakage of sodium out of this axon is greater than it is in this axon with a large radius. That reminds me of something I said earlier in the class. But let's see, let's have some fun here. Michael, how about you? Why would that be? Why is that important?
It has to do probably with surface area to volume ratio.
Okay, all right. Mandy, where's Mandy? Mandy, why is that important? Okay, that's all right. Let's see here. Andrew, where's Andrew?
Yeah.
Andrew, what would you say?
The larger one will have a overall smaller surface area than compared to the thin one.
Okay, all right. Miss Camilla, we're not going to ask you a question tonight. You were battle tested on Monday night. You thought your heart just started going. All right, let's see. Jocelyn, where's Jocelyn? Jocelyn, you're up to bat. Okay, so it's fair. Kent Wynn. Where's Kent?
Can you repeat the question for me?
Sure can. The conduction velocity is higher in that larger diameter or larger radius axon as compared to the smaller one. There is no myelin, so we're just talking about why is it that the conduction velocity would be faster in that larger one?
Is it because the radius of the axon is larger? And
But why is that important? That's the question. I'm glad actually you said that, is because that's the question, is why is that radius so important? And it turns out, yeah, go for it.
I was going to say, I was thinking it's because if it loses less, if the smaller radius axon loses less sodium in general as compared to the larger axon, then that means the action potential is slightly more insulated, but not, it's not myelinated, so it's not insulated, but it loses less, so the action potential gets propagated more consistently.
I think what you're saying is that Michael and Andrew had the right answer. Yeah? It's a property of surface area to volume ratios, right? In the smaller axon, the surface area is proportionally greater than it is in the large axon. And therefore, our current leaks out, more of our current for any distance leaks out here as compared to here. And basically you said that it would kind of get further down the axon for any given depolarization, and that's exactly right. So I'm going to continue to try to emphasize the surface to volume ratios, because this is not the only place where that applies. Thinking about thermal regulation is not the only place it applies. Anytime we're dealing with something that involves a surface area and a volume, you got to be thinking of the potential importance of this ratio. Yeah, go for it.
So the,
Oh, now you want to talk. You didn't want to talk a minute ago.
just to clarify, so with the smaller axon, there's more room for it to leak pretty
the action potential will be conducted further down the length of the axon. And we're gonna have greater leakage or conductance of sodium out in the smaller one. All right, so I just wanted to make that point to you. Make sure that you appreciate the importance of both of these, myelination and the radius of the axon. And for the radius of the axon, you're gonna sit there and say, this is what is referred to as the cable property. I don't want you to necessarily to know that. If you do, great. But that cable property revolves around simply a surface area to volume ratio. And when the surface area to volume ratio is greater, that means that we have greater leakage of sodium out of the cell. All right, so coming back to where we left off on Monday night, just to kind of repeat a bit of what I stated on Monday night. Let's say that we have a receptor-gated sodium ion channel. So in this hypothetical, we're releasing a neurotransmitter that will bind to the ligand binding site of this receptor. And when that occurs, we're back to first night in this class. When that neurotransmitter binds to that receptor, we're talking about which principle from night number 1. Mr. Corey, you're the expert on this, seemingly. This is a story of homeostasis, right?
No. This is how we can speak structures to its function again.
It is, that's exactly what it is, because the binding of that ligand to the receptor alters the structure of this protein that happens to have an ion channel too, and now the ion channel transitions from a closed to an open state. And in so doing, you're going to tell me that sodium will flow into the dendrite of this postsynaptic neuron according to its electrochemical gradient. Well, we've got positive charge coming into the cell, and that's going to cause this cell to hyperpolarize, to cause it to become more negative, right? If I got positive charge coming in, the cell's going to become more negative. No, it'll become less negative. In other words, it will depolarize. So our membrane potential will now move from minus 70 millivolts, and if we get enough positive charge coming into the cell, and we can get the membrane of this get the axon of this postsynaptic neuron to threshold, then all the voltage-gated sodium ion channels in the axon of our postsynaptic neuron are going to open up, right? And we're going to have depolarization down along the length of the axon of this postsynaptic cell. In this instance, when we've got positive charge coming into the postsynaptic cell, we would say that this represents what's referred to as an excitatory postsynaptic potential, the so-called EPSP. And I had mentioned that to you on Monday night. In dealing with ions like potassium, so if we say that this is a receptor-gated potassium ion channel, here we're going to cause this potassium ion channel to open up, and there's going to be a relatively weak efflux of potassium out of the cell. That's going to cause in the side of this cell to depolarize. Right? No? Are you shaking your head yes or no? It looks like you're saying yes when you're doing that. What is your answer? The cell going to depolarize, stay the same membrane potential, or hyperpolarize? That's right, it will hyperpolarize. And here's the next question. If this cell hyperpolarizes, are we moving closer to threshold or further away from threshold? Or further away from threshold. Is this cell going to be more excitable or less excitable? It's going to be less excitable. We're going to need a much stronger stimulus to get us to threshold. And therefore, we're going to say that when we've got positive charge leaving, or if we had negative charge coming in, that would create not an excitatory postsynaptic membrane potential, but an inhibitory postsynaptic membrane potential. Any comments, questions, thoughts, confusion about that? Don't be bashful to speak up.
Can I just try to explain the potassium one to see if I have the concept correct?
Sure.
So when a chemical messenger binds with the receptor of the receptor-gated potassium ion channel, then it's going to open up and then the potassium is going to flow out, right? Which would
It's going to flow out
according to its electrochemical gradient.
Okay,
And
and the
it would
the
cause
chemical gradient favors what? The efflux or the influx of potassium?
the efflux.
That's correct. The electrical gradient favors,
going inside of
okay.
the
And
cell.
so which of those two forces, so I've got opposing forces, which of those two is strongest?
The chemical?
That is correct, because we know that the membrane potential is not at the equilibrium potential or even more negative than that.
For potassium.
And therefore, the chemical gradient is stronger. Is it stronger by a lot or a little bit? And the answer is, it's stronger by a little bit,
By a little bit.
right? Because we're close to the equilibrium potential for potassium, but we're not there.
Okay, yeah. Okay, that makes sense. And because the equilibrium potential is 93, negative
Minus 90 millivolts, minus 92. It doesn't, ballpark, minus 90 millivolts.
And so this would cause the cell to hyperpolarize because it's becoming more negative as
We're having
potassium
positive
leaves.
charge leaving, which is going to cause the inside of the cell to become more negative.
Okay, that makes sense. Okay, cool.
Good. What do you think? How'd she do? I think she got 2 thumbs up. So, one of the things that I want to highlight for you is that at the dendrite, we do not produce a action potential. We produce a graded potential. So, we're talking about action potentials. Action potentials are properties of voltage-gated ion channels. And for us, we're just gonna say generically speaking, that action potentials are properties of voltage-gated sodium ion channels. And these, we're gonna say, are all or none events. Okay, where do action potentials occur? You're going to sit there and say, anywhere where I've got voltage-gated sodium ion channels. And as it turns out, and again, I just want to highlight this for you and emphasize this to you, make sure you understand the distribution of key membrane proteins along the length of the neuron, because that comes back to night number 1 in here, principle number 1, structure determines function. If there are no voltage-gated sodium ion channels here, then this region cannot produce an action potential. If there are no voltage-gated sodium ion channels here, that can't produce action potential. There are voltage-gated sodium ion channels and potassium ion channels all along the length of the axon terminal, and there we're going to be able to find that that membrane can produce action potentials because of the structure, meaning the presence of voltage-gated ion channels. Here we have receptor-gated ion channels. And the amount of charge that we allow into or out of that cell is dependent upon how many of these receptor-gated ion channels have a neurotransmitter bound to them. So let's just say that there's 1,000 voltage-gated sodium ion channels here. So the hypothetical let's say that we've got 1,000 voltage-gated sodium ion channels. Don't want this. Forget that. Sorry about that. Too much. Luckily, I catch these. Hopefully, I catch them. Oh, geez, come on. There we go. We've got 1,000 receptor-gated sodium ion channels. Let's say only one of them opens up. Is there going to be a high conductance of sodium into the dendrite or a very low conductance? Low. Are we going to see change in membrane potential? Yeah, if we had a really, really super sensitive system, we'd see a little upward blip, but that's it. If we got 200 of these receptor-gated sodium ion channels to open up, would that produce a maximal change in the membrane potential? No, it would only produce about 20% of it, the maximal response we could get. And so the point I'm getting at is here at the level of the dendrite, we don't have action potentials because we don't have voltage-gated ion channels. We have receptor-gated ion channels. And the change in the membrane potential is dependent upon how many of those receptor-gated ion channels are opened up. It's not a all or none event at the dendrite. It can vary from a few to a lot opening up. What do you think? Everybody with me on this? And so the response that we're going to produce here as a result of these receptor-gated ion channels is graded. It can be small, it can be medium, it can be large. It can't be all or none. So over here under graded potentials, we're referring to these as graded potentials simply because the number of ion channels that opens up is dependent upon how much neurotransmitter is released and bound to our receptor-gated ion channels. Here I'm writing ligand in parentheses, I'm going to put NT for neurotransmitter. All right, let me pause for just a moment and ask if there's any questions, comments, confusion that we need to clarify. Do not be bashful to, yeah, go for it.
I don't remember where the soma fit into the diagram of where things are what, so receptor-gated ion channels are found on the dendrites and the soma as well?
We're gonna keep the story really simple, because everything I'm saying has got variations associated with it,
Okay,
but we're just gonna be, like I said, very simple.
so for the sake of it, dendrites?
For our purposes right now, if you wanna go get a master's degree or a degree in neurophysiology, you'll be able to learn about all these variations, because everything that I'm describing is just kind of the basic principles,
Okay,
but there's always variations.
okay.
Yeah. All right. Now the system I'm describing here that involves receptor gate ion channels, we're going to refer to that as being ionotropic. So here, I'm just going to come along. No matter whether we're producing inhibitory or excitatory postsynaptic membrane potentials, we're going to refer to any system that involves a receptor-gated ion channel here as being ionotropic. And one of the properties of these ionotropic systems is that they respond relatively fast. So here I'm just going to put fast response time. What that simply means is that these receptor gate ion channels are going to open up quickly, and they're going to allow whatever ion they're conductive to move through quickly. That's all that means. And now I'm going to show you a different system. And to show this system, I'm going to change my depiction or my diagram a little bit. And so now I'm showing the dendrite, and I'm representing the membrane of the dendrite here as a phospholipid bilayer. In the previous drawing, I represent the phospholipid bilayer of the dendrite simply as a single line. But for the next part of the story, we need to represent the membrane here where you can see the lipid bilayer of it. So here's a receptor, and you may have seen this. I hope you've seen this, because if you haven't, then come see me afterwards. So here's the receptor. And associated with this receptor is a G protein. Here I'm going to put GS. Hopefully all of you sitting going, oh, okay, he's talking about some type of a second messenger system. Here's our amplifier. Here I'm for, just to provide some specificity, I'm going to put capital A, capital C, adenylyl cyclase. So here comes our neurotransmitter, and it binds to this receptor. That receptor's structure is changed. So now it can act on the stimulatory G protein. And acting on the stimulatory G protein, its structure is altered. the alteration, the change in the conformation of our G protein then leads to a change in the conformation of our amplifier or this enzyme, which is going to come along and it's going to convert ATP into cyclic AMP. Listen to how long it takes me to explain this story, and then I want you to rhetorically come up with the answer. Is this a fast process or a slow process as compared to a receptor-gated ion channel? All of you are going to tell me that this molecule cyclic AMP is going to act on a inactive form of a protein kinase. I'm just going to put here IPK. And that's going to convert this protein kinase from its inactive form to its active form. And over here in the membrane, we've got a calcium ion channel. And what do protein kinases do? They phosphorylate their target molecule. Then we're going to say that ultimately this protein kinase in its active form is going to act on this calcium ion channel in a positive fashion, and that's going to allow calcium to come flowing into the cell according to the equilibrium, according to the electrochemical gradient for calcium. And that electrochemical gradient for calcium is very high, right? Because first of all, there's a 10,000-fold concentration difference, and the equilibrium potential for calcium is plus 120 millivolts. Inside the cell is at minus 70 millivolts. So we have a very strong electrical gradient that favors the influx of calcium. We have a very strong chemical gradient that favors the influx of calcium. Calcium's coming in very quickly. But this process involves a number of steps before we can get that ion channel to open up. And therefore, you would tell me that this system, relative to a receptor-gated ion channel, is faster or slower?
So,
And the answer is slower. This type of system is not referred to as ionotropic. This is referred to as metabotropic. And this involves some type of second messenger system. Now, one of the things I'll say, and hopefully what I'm going to say is 100% correct, is that what is behavior? And how many of you learned about Sigmund Freud? Anybody in here? Okay. Gestaltism. I'm glad you haven't, actually, because this is where things were when I was coming through, is that Sigmund Freud was the huge figure in psychology way back when. And there was no true understanding of behavior. It was just kind of like, yeah, philosophically I think this or I think that, blah, blah, blah. And for me, as a student back in 1970, when I took my introductory psychology class, it was just like I was listening to a bunch of BS. And I think I may have told somebody the story. And I came home and I was telling my dad about this, and I'm expecting I'm gonna get a sympathetic ear. And he sits there and says, you just gotta take the class, you just gotta get through it and get on with it. And that's what I did, but it was a struggle. Did well in the class, but it was a struggle just to listen to this gobbledygook every night. Hopefully I'm not telling you gobbledygook every night. But at that time, I was sitting there going, there's gotta be some type of biological explanation for what we refer to as behavior. And at that period of time, the field of psychology, unknown to me because I'm just a novice, was undergoing a huge transformation because there were these neurobiologists, there were psychobiologists who were sitting there and telling the rest of the behavioral community, hey, we need to understand the biology of behavior, not just coming up with all these whimsical ideas. And so from this, you can come to campuses like UCLA, any of the top campuses in the country, and you're going to see people who are neuroscientists, neuropsychologists, physiopsychologists, who really are trying to understand the biological basis of behavior. And in the brain, every neuron's got input from about another 1,000 neurons. So good luck in trying to understand behavior. Incredibly complex. But at the bottom line of the story is behavior is dictated in large part by the types of neurotransmitters that are being released and the receptors to which they're binding on the postsynaptic cell. And in thinking about behavior, we haven't come all that far in terms of understanding the underlying biology of behavior. But we do understand the more extreme forms of behavior. For instance, schizophrenia. Schizophrenia, I would say, is truly an extreme form of abnormal behavior. Come to find out, there's a receptor for dopamine, which is a neurotransmitter. And there are different types of dopamine receptors. There are DP1s, DP2s. What I've just described for you is like a DP1 or DP2 dopamine or dopaminergic type system. And it turns out that people with schizophrenia have an abnormal localization of DP2 receptors. So there you can go in, you can actually study the localization of these DP2 receptors in an individual's brain, and you can determine, okay, yeah, they do have this abnormal localization in numbers of this DP2 receptor that then puts them at a much higher risk for the development of schizophrenia. The other thing I would say, sadly, is that when I was a kid growing up, when you talked about drugs, I think you're probably talking mostly about alcohol or amphetamines. That was it. Nowadays, there's a whole potpourri of drugs out there. And sadly, in some individuals, these drugs will mess you up for the rest of your life. Crack. How about crack? Crack supposedly is one of the most addictive drugs out there. Once you've had one experience with it, you're chasing the so-called dragon to try to get the same high that you got that initial time. And sadly, those people, man, talk about a change in their lives. That's a terrible thing to go through. Sadly, there was an article out about this granddaughter, of this famous singer. You guys probably never heard of him. His name's Elvis Presley. Maybe you hear of Elvis Presley. Come on, raise your hands. Just humor me. All of you have heard of Elvis Presley, right? And she got hooked on drugs. She said she was taking something like 80 drugs a day. I forget what pill she was taking. But sit there and think about that. Holy mackerels, that's sad. Truly sad. Okay, so make sure you understand the difference between ionotropic and metabotropic types of systems. Now I want to talk about, come back to graded potentials, and we're going to talk about integration. So this is just going to represent a neuron. And as I said a little bit earlier, in the brain, neurons are getting information from a lot of other neurons. The blue guys are going to be excitatory. And when I say excitatory, what I mean is they're going to produce an excitatory postsynaptic membrane potential on our postsynaptic cell. And so in parentheses, I'm just going to put a positive sign in blue for each of these, and you're going to understand that that means that they're going to produce EPSPs on this postsynaptic cell. And we can have other neurons that are going to produce inhibitory postsynaptic membrane potentials, and I'm just going to represent those in red and using this negative sign in parentheses. And then we're going to create a graph. And on the y-axis, we're going to put membrane potential, so I'm just putting capital D, subscript M. Make sure you understand that the units here are in millivolts. And our axis is going to start at minus 100 millivolts and just go up in increments of 50 millivolts. Threshold. We're going to say that threshold, we're using this textbook value of minus 40 millivolts. And we're using our standard textbook value for the resting membrane potential of using minus 70 millivolts. All right, now, there are different ways in which we can depolarize our postsynaptic cell. And I'm going to describe these two different ways in which that can occur. So here's the first example, is that we've got our postsynaptic cell here. We've got it at minus 70 millivolts. Oh, let me go ahead and number these also. So I'm going to number our cells here. The blue ones I'm just going to number as 1, 2, and 3. So we come along, and at this point in time, cell number 1 releases some neurotransmitter. And when that happens, we get a relatively small depolarization in that postsynaptic cell. So you'd sit there and say, we didn't produce an action potential. Well, we try a different strategy. And that is, we come along and we stimulate cell number 1 2 times in rapid succession. So we get a response that looks something like this. Remember the term that I used. These are graded potentials. Graded, they can vary in size depending upon how much neurotransmitter we are releasing. And again, you would tell me, well, Vince, we didn't get to threshold, so we're not going to produce an action potential in that postsynaptic cell. So we come along and we employ a different strategy, one in which I think all of you would predict, having seen the second example. And here, we're going to stimulate cell number 1 3 times in rapid succession. And so this is going to cause, we'll say, 3 times as much neurotransmitter to be released. We're going to have 3 times as much receptor-gated ion channels that have got a ligand bound to it. And here, using this approach, we end up producing a so-called, well, we end up producing an action potential in that postsynaptic cell. So we're talking about integration. Integration is occurring here in the synapse between the pre- and postsynaptic neurons. And we're talking about the different strategies that can be employed to get to threshold. And so the strategy is here that we varied the frequency at which a single neuron fired. And so this is referred to as temporal summation. And this word temporal just give me 1 second here. I need to get something to drink real quick, if you don't mind. That word temporal means with respect to time. That's frequency. Frequency is the number of times, is the number of events per unit of time. And again, I like to have lots of fun in here to compare generations from one to another. And in my generation, we grew up with a phenomenal show. And I know none of you've seen this, but you should go see it. It's called Star Trek. Anybody in here ever hear of Star Trek? I'm being facetious. Captain Janeway, Jean-Luc Picard. Who else was there? Anyway, who? Yeah. Yeah. Yeah, and one of the cool things is they even had a god in that. They had the God series. What were they called? The Qs. I'm not sure why they used the Q, letter Q for a godlike being, but it was kind of cool. Anyways, in Star Trek, here's what they did. They had a lot of temporal events related to time. They went back in time. They went forward in time. And they had a lot of spatial events. Being in space here, one moment, being in space over there, the next moment. So in talking about summation or integration, we're talking about temporal events, and we're going to talk about spatial events. And this is not in relationship to space there, it's in relationship to space over there. Yes, sir.
Can you define integration again?
Yes, integration is the process by getting all this input from all these neurons, and somehow that's talking to this neuron here. Right? Somehow that postsynaptic neuron's got to be able to make sense to all the information that's coming to it. And so, in thinking about that, we're thinking about temporal summation. So here we're talking about the frequency at which a neuron or neurons are firing. But there's a different strategy that we could use. The result is the same, but the approach is different. So we'll just come back and use a similar type of discussion. We've got neuron number 1. It fires and it releases a certain quantum, a certain amount of neurotransmitter that binds to a certain number of receptors, which then causes a slight depolarization in our postsynaptic neuron. We didn't get to threshold, so we're not going to produce an action potential. So we come along and we try a different strategy. And in this strategy, neuron number 1 and neuron number 2 are releasing neurotransmitter onto that postsynaptic cell. And because we're releasing more neurotransmitter, we're going to depolarize the cell more. In our hypothetical, we did not get to threshold. Therefore, we're not going to produce an action potential in the postsynaptic cell. And you guys are going to tell me that last one there, that I'm going to go up there and I'm going to say that neurons number 1, neuron number 2, and neuron number 3 are all firing at the same time, and they're adding the amount of neurotransmitter that each of them is releasing adds onto one another, causing more receptor-gated ion channels to open up. And that gets us to threshold, and so using this type of approach, we're able to generate an action potential in the postsynaptic neuron. And so here, as I said, we're not going to refer to this as temporal summation. This is an example of spatial summation because we've got different neurons in the space. Spatial meaning space around. We've got different neurons contributing to getting this cell to threshold. And so this is referred to as spatial summation. I need my Trader Joe's cookies right now. Questions? Don't be bashful, speak up.
What kind of cookies?
Don't do that. My diet's bad enough. All right. Just give me 1 second. Yes, go for it.
I'm sorry, just to make sure, so integration has either a temporal summation or a spatial summation.
Yes, that's correct.
And the temporal summation is the first example where we only have the input from one neuron, and the spatial is when we have the summation of multiple.
Yeah, so, but what you have to appreciate is that when you talk about temporal summation, we're talking about the frequency. Frequency is really important. That's the events per unit of time. So we could stimulate a cell once. We could stimulate a cell. So here's my stimulus. Say pop, pop, pop. That's a long period of time between each of those stimuli. Here's a different approach. Pop, pop, pop. That's temporal, right? The stimulation is occurring at a much higher frequency. Frequency is the number of units per unit of time. How many stimuli per second or per millisecond? All right. So now I'm gonna go ahead, I'm gonna erase this. Everybody's good with the information up there, hopefully. So I'm going to come along and represent now the central nervous system just using a box. So CNS, that's just an abbreviation for central nervous system. So here we're talking about the brain plus the spinal cord. And we can have output from the brain and the spinal cord, or we can have input into the brain and spinal cord. And what we're going to talk about are different types of neurons and sensors and things of that sort. So over here, I'm going to show a blue line and in this box I'm going to put P, N, S, and that stands for peripheral nervous system. And then underneath the peripheral nervous system, we're going to show two branches. There is the somatic branch. There's that word soma. What does the word soma mean? Anybody? Body. And if you sit there and think about the human body, the majority of our mass is due to the presence of what organ system? Say it louder. Skeletal muscle, right? So when we talk about the somatic branch of the peripheral nervous system, we're really talking about the innervation of our skeletal muscles, the neural input into our skeletal muscles. Well, there's another branch underneath the peripheral nervous system, and this is the autonomic nervous system. Here I'm going to put capital A, capital N, capital S. And the autonomic nervous system controls a whole chunk of our physiology / biology. Really plays a huge role in regulating so many different aspects of our physiology. And the autonomic nervous system consists of two branches. There's the sympathetic branch. And then there's the parasympathetic. So here I'm going to put efferent. And then there's the input. And this will be the afferent side of our equation. And we're going to say that there are three different types of afferent input. The first we're going to refer to as somatic senses. That was good. The somatic senses: skin, touch. Skeletal muscle. Sit there and think about how we take our ability to move for granted. All of you can sit there, close my eyes. I just want to move my hand from here over to here. I did that. How the heck am I able to do that? And the answer is because we've got all these sensors. And in our muscles, there are length sensors. And those length sensors are providing feedback to the brain to tell the brain where my hand is in space. On top of it, there also are sensors in the joints throughout our body. And those sensors are also helping. There's what are referred to as special senses. Here we're talking about vision. Hearing. Taste. Vision, hearing, taste. What else am I leaving out here? Smell, and also, oh yeah, how could I forget this one? Here's an interesting one. I was talking to somebody about this just the other night. Equilibrium. What am I talking about when I'm talking about equilibrium? Your ability to stand upright and know spatially where the body's at. That comes from the ears. Within the ears, I'll talk about this downstream. There are these sensors that help the brain figure out where we are in space. For spatial orientation, there's 3 key sensors. Eyes, the eyes tell you where you're at. We've got sensors in the ears that basically are looking at X, Y, and Z in terms of the space. And then we have what's called seat of the pants. Like right now, believe it or not, there are pressure sensors providing feedback to the brain center going, hey, he's sitting down, he's upright. Sometimes what happens is that the brain is integrating all this information. It's taking the visual cues, it's taking the cues from the ear, and it's taking the seat of the pants, and it gets confused. And when that happens, guess what? Your equilibrium changes dramatically, where you may feel like you're gonna fall. And then we've got the special senses, and then we've got so-called visceral. And the visceral senses. We just talked about this, visceral senses, feeling full. So the gut providing information back to the brain center saying, you know what? We don't need any more food. I'm just going to put fullness. The regulation of pH. And what else? There's one more. What am I forgetting here?
Temperature?
What's that?
Temperature? Would temperature be a visceral sense?
No. But thanks for saying that. Blood pressure. How could I forget blood pressure? Okay. So this is an overview. And now what we're going to do is we're going to focus in on this part of the story. Can I take you guys another 10 minutes or any protests or not my good another 10 minutes? Okay. All right. So this diagram I'm going to develop for you is going to be a base diagram. We're not going to finish it tonight. We'll finish it on Monday, but there's a lot of info in here. And we're just going to say that what I'm showing is the brain and the spinal cord. What I'm going to show to you is basically a concept model. What I'm showing you is not anatomically correct, but it's a conceptual model. And what I'm going to describe for you is a motor neuron. Motor neurons innervate our skeletal muscles. I'm going to describe for you a sympathetic neuron. And I'm going to describe for you a parasympathetic neuron. And there will be similarities, but there also will be differences between each. So here's the scheme. Our motor neurons are found in the cell body of the motor neuron is found in the spinal cord. And as a matter of fact, if you were to look at a spinal cord the human spinal cord, you'd see a bulge in the thoracic region, and you see a bulge in the sacral region. And those bulges are there because that's where the highest proportion of motor neurons are located. The density is high in each of those areas, causing this bulge. So here's the cell body of our motor neuron. And here's the axonal projection of this motor neuron. And I'm not going to do this down the entire length. Of the neuron, but you're going to interpret this to simply mean that the axons of motor neurons are heavily myelinated. So, two important attributes for us is that the axons of motor neurons are heavily myelinated and they have a very large radius. You'd sit there and say, wow, okay, both of those mean that these motor neurons have a very high conduction velocity for the action potential. Down here, I'm just going to put a box. And above that, I'm going to put target cells. And because we're talking about skeletal muscle, it's simple. The target cell of a motor neuron is skeletal muscle cells. By the way, these are also referred to as muscle fibers. So I'm just going to write the words muscle fibers. So an individual skeletal muscle cell is a muscle fiber. Okay, now if I can have everybody's attention up here. What I'm going to do in each of these examples is I'm going to write the name of the neurotransmitter above this. Hopefully all of you would interpret this to represent the axon terminal. But I want to be very cautious here with you. So in the scheme I'm laying out, the neurotransmitter is going to go on top, and the neurotransmitter receptor, which is actually here, is going to go on the bottom. Everybody with me? It's just a scheme I'm going to use. Everybody with me? So just make sure that you understand on the bottom, I'm going to have the name of the neurotransmitter receptor, but that receptor is not here. It's here. It's on the membrane of our target cell. Okay? So, what's the name of the neurotransmitter? Is acetylcholine. I'm just going to put capital A, capital C, lowercase h. That stands for acetylcholine. What's the name of the receptor? The receptor is an acetylcholine receptor. I'm just going to put capital A, capital C, lowercase h, capital R. And you read that as an acetylcholine receptor. However, there are different types of acetylcholine receptors. So there are so-called nicotinic ACH receptors, and there are muscarinic ACH receptors. Why is one called a nicotinic ACH receptor and another a muscarinic ACH receptor? Because if you come along and you drop nicotine onto a nicotinic acetylcholine receptor, its ion channel will open up. If you drop muscarine onto a nicotinic ACH receptor, its ion channel will not open up. So there are different types of acetylcholine receptors, and the type is named after the type of molecule that gets them to open up. So here I'm going to put a small little N. And that means that it is a nicotinic. You guys have all heard of nicotine, right? Yeah, no, maybe so. Nicotine, a highly, highly, highly addictive drug, not just slightly addictive, but highly addictive. One of the most addictive substances out there. I'm going to do one more, and then we'll call it quits here for tonight. So here's a sympathetic neuron. So the cell body is located within the spinal cord. There's a short axonal projection. And then there's a postsynaptic cell. And it's got a relatively long axonal projection. Just give me a moment here. Sorry, gotta modify this a bit. Okay. So there's this synapse, and oh, by the way, there's this cluster of neurons immediately outside the spinal cord. What do we call clusters of cells? Clusters of neurons. Fantastic, very good. So there's this so-called dorsal root ganglion. Excuse me, not dorsal root ganglion. I'm just going to put ganglion here. And this ganglion is a cluster of cells. So what's the name of the neurotransmitter that's released by this presynaptic cell? And the answer is it's acetylcholine. What's the neurotransmitter that's found on the dendrites of the postsynaptic cell? It's a nicotinic ACH receptor. What are our target cells? Target cells, cardiac muscle. Smooth muscle. And various glands. What's the chemical messenger that's being released by this postsynaptic cell or the sympathetic neuron? And the answer to that is it's norepinephrine. So I'm just going to put capital N, capital E. That stands for norepinephrine. Now, there's norepinephrine, there's epinephrine. Epinephrine goes by a different name, adrenaline. Norepinephrine goes by a different name, noradrenaline. So what are we going to call the receptors located on our target cells? And we're going to refer to those as adrenergic receptors. All right? And there are different types of adrenergic receptors, which leads to a lot of fun biology. So there are so-called alpha-1 adrenergic receptors. There are alpha-2 adrenergic receptors. There are beta-1 adrenergic receptors. And there are beta-2 adrenergic receptors. Just talked about this briefly before, but let's just see it, and you don't have to respond to this if you don't want to. Anybody in here who uses a rescue inhaler? Anybody who's asthmatic? Okay, so we talked about this before. In the medication you use, I'm assuming you use a rescue inhaler, nebulizer, some type of device. And so you take that to your mouth, and you do a little puff, and you breathe in this gaseous mixture. And in there is a drug known as, what's the name of the drug? Starts with an A? Albuterol. Albuterol is a beta-2 agonist. Beta-2 agonist, what's he talking about? Right there. It's going to act on beta-2 adrenergic receptor. The heart, you work in an animal facility, right? Do you guys treat any dogs or cats with any heart disorders?
Yes.
And what type of drugs do you use? Do you use anything like Enderol, propanolol?
Furosemide, Trimabendin.
Okay, she uses furosemide. Furosemide is acting in the kidneys. So those are that's a diuretic. But we can use drugs to treat the heart using beta blockers. Beta blockers, you guys are going to learn a little bit about that. Beta blockers are acting primarily on beta-1 adrenergic receptors. Anyways, there's a lot of our physiology that's regulated by this class of receptors right there. And what you're going to learn is that each one of them is a different type of transduction system. Some are going to involve cyclic AMP. Some are going to involve IP3. Some are going to inhibit the production of cyclic AMP. Some are going to act on inhibitory G proteins. But the cool thing is that based upon what you already know, come Monday night, you're going to begin to learn about the transduction systems of each of these, and that theme is going to continue throughout the remainder of this course. Okay, so I've said enough, and I apologize for going over. When we come back on Monday night, we're going to pick up the story. We're going to talk about parasympathetic neurons and the types of neurotransmitters that they're using and the receptors that they're acting on. And then we'll get into more depth in terms of what each of these involves here. So we can have the same chemical messenger, but produce entirely different types of cellular responses because of differences in the transduction system. So imagine the same chemical messenger can produce entirely different responses based upon the transduction system involved. Really cool biology. All right, I'll see you guys upstairs, we'll say in about 15 minutes. If you have questions, comments, thoughts, please don't be bashful, come on up.